Wait, they did what?
A slab of 3D printed plastic, 3 millimeters thick. Sound shines through it and arranges living human cells into tissue in seconds. And in May, those cells did something extra. Kang, Jeong and Han reported in Nature Communications that sound-arranged cells ran their own development. The plate works like stained glass for ultrasound. Every point, a different thickness. Melde and Fischer printed the first in 2016. Who funds it now is the odd part. That first Max Planck plate cost about the price of a dinner and rebuilt Picasso's dove in floating powder. By 2023, whole 3D cell shapes in one shot. Extrusion bioprinting squeezes cells through a nozzle for hours. Shear kills them at the density real tissue needs. Sound places every cell at once. Here's the part I didn't expect. The NSF awards that surface for this field total 1.35 million, and none of them touch acoustic bioassembly. In Davos, Tiziano Serra's group uses these patterns for the tendon-to-bone interface no surgeon can stitch. We took that apart in the podcast. Rotator cuff repairs fail at rates reported from 20 to 90% because that gradient can't be sewn. Sound can draw it in seconds, in a dish so far. In Bristol, James Armstrong aligned muscle precursor cells with standing waves until they fused and contracted. Acier Marceau's open levitator cost $70. Over 100,000 Americans wait for organs. Up to 17 die daily. The near-term use is smaller, human tissue models, as the FDA walks back animal testing. The catch is plumbing. Nobody has built capillaries at 10 microns, and the know-how sits in maybe 4 labs on 3 continents. Whether this stays cheap or gets locked into exclusive licenses may be decided within 30 months. We hedge what the sources hedge. Full report, KPsphere.com.
See you next time.